This is TWIS. This Week in Science, episode number 989, recorded on Wednesday, October 9th, 2024. Who are the science winners? Well, I'm sure we're going to find out. I'm Dr. Kiki, and tonight on the show, we will fill your head with prizes, pessimism, and potential pathways. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. As another hurricane is hitting the people of Florida, a storm so intense that it has caused the formation of several tornadoes ahead of reaching land, some as large as 2 miles wide. The folks here at This Week in Science hope that everyone will keep safe, keep dry, and get out of the area in time. But we know that many will not. Even with a history of dealing with large storms, we have learned very little about how to prepare, how to move millions of people to safety with relatively little notice. I want everyone to pay close attention to events like this. It is not normal. Hurricanes are 3 times more frequent than 100 years ago. The major category storms, 3 or above, have doubled since 40 years ago. The worst storms in recorded history are all happening now. The highest heat waves, the biggest hurricanes, now with tornadoes. As you see this happen, and see it happen with more frequency, ask yourself, why? And if you know, you know. This is not a test. This is This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
And a good science to you too, Justin, and everyone out there. Welcome to another episode of This Week in Science. We are back again as we missed last week. I apologize to those of you who are looking forward to another episode, but events conspired to keep us away from our live stream and from our podcast last week. But we're back now because we always love to be here. And so we have this wonderful episode all for you right now. Thank you for joining us. I brought stories, of course, about the 2024 Nobel Prizes in Science because it's that time of year. Award time. Bird feet, comb jelly combinations, brain development and deterioration, and a chapter to close, perhaps. What do you have for us, Justin?
Oh my goodness, what do I have to start the show with? I've got stressed bees.
No way.
The benefit of less active running. A galaxy that should not be. And plunging biodiversity.
Plunging, like with a plunger? You're plunging biodiversity.
Actually, yeah, more like with a plunger.
Oh no, okay. All right, well, we're going to have a great show. It's not plunging at all. It is going to be exciting. And you know, this show will be full of science story winners. Winner, chicken dinner. Okay. As we jump into the show, I want to remind you that subscribing to the TWIS podcast on your favorite podcast platform, YouTube or Facebook or Twitch, will bring you TWIS each and every week, every time a new episode is published. You can watch live on our live stream channels Wednesdays, 8 p.m.-ish Pacific time until we finish the show. Sometimes it's a tight 90. Most often it's not. But anyway, whatever you need, you can search for This Week in Science or just twis.org in your browsers. Now it's time for the science. Oh, yeah. Are you ready?
Yeah.
Yeah. Okay, we're going to do our celebrations right up at the top of the show. It's time for that wonderful time of year where we celebrate Alfred Nobel and his statue. Actually, that's not what it's about at all. The celebration is meant to celebrate discoveries that are really impactful to humanity, that have changed the way that science, and not just science, but society operate together. The Nobel Prizes in 2024 are as follows. Dun, 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 dun. In physics this year, John Hopfield and Geoffrey Hinton, or if you go with the British pronunciation, it's Geoffrey Hinton. These two researchers have used physics to find patterns in information. Machine learning. Oh my goodness. Yes. So, John Hopfield, quote, for foundational discoveries and inventions that enable machine learning with artificial neural networks. Geoffrey Hinton, for foundational discoveries and inventions that enable machine learning with artificial networks. Isn't it great? Amazing. The same thing. I mean, I think they work together. These researchers were brought into the Nobel celebration this year for the influence that their work has had on modern AI and the development of the tools that we use in current day to, I don't know, for computers to take over the world. No, for everything from discovering protein function, protein architecture and structure, to helping you work on your essays for your college classes. The machine learning that is involved is based on these artificial neural networks. They use large language models and many other types of models that together sort and analyze data based on probabilistic matrices and come up with information that sometimes hallucinates, but that's today. Maybe tomorrow it'll be even more accurate. But I don't know, Justin. I think that machine learning, where once it was very simplistic, now it is giving us insights into the human brain. And it is also just, there's no way we can stop it from influencing humanity's future.
Well, yeah, certainly. You mentioned the protein research. That has been massively accelerated already by AI, constructing all the possible protein configurations, then being able to also pick an interaction that it wants the protein to have and back engineer and design a protein based on that. It is really a huge change in the way that research doesn't have to do a sort of form of guess and check as much. Now it can have a menu from which to choose to attempt at least to get the interactions it wants. That's going to be a massive change.
It will be massive. And whereas you can just look at a network or a machine learning as just math, these Nobel laureates use tools from physics to lead to the techniques that we use today, the models that we use today. So John Hopfield created an associative memory that can store and reconstruct images and other types of patterns. Geoffrey Hinton invented a method that can autonomously find properties in data. And so it can perform tasks like identifying specific elements in pictures, like fingers, for instance, fingers on hands. No. I think machine learning, though, is the learning side of it is the interesting aspect, because the way that you put their models together, John Hopfield used what is called the Hopfield network, and it uses physics describing a material's characteristics due to its atomic spin. So each atom is a little tiny magnet. And so the energy in the spin it all works together. And it's the way the nodes work together. And so if you have a really, really high quality image, then the image is going to have the values so that the whole image has a lower energy level because they all balance each other out over time because of the way that their quote unquote magnetic fields would be connected to each other. Geoffrey Hinton used the Hopfield network, but then used the Boltzmann machine to recognize characteristic elements in different types of data. So whereas there was images used in Hopfield stuff, Hinton used tools from statistical physics, and this is it was trained by feeding examples that are very likely to arise when a machine is run. So classical physics, Boltzmann machines. And so this has been built upon and built upon and built upon and will continue to be built upon again and again and again. Let's move on to chemistry. Yeah.
Yeah.
The Nobel Prize in Chemistry. We have interviewed one of the Nobel Prize winners in chemistry. Prior to COVID-19, and even prior to that, we spoke with a researcher named David Baker. The Baker Lab at the University of Washington in Seattle has been doing amazing work in computational protein design using machine learning models for a very, very long time. We've talked about things like Folded at Home and the Rosetta model and various others that have been created and released into the wild by the Baker Lab. The Baker Lab is responsible for countless discoveries using their models to be able to take from what is understood, what is seen, and then create new proteins, new structures, new functions de novo. Also to model and copy. And it is a part of our journey in understanding how biology works. And so I'm extremely excited that David Baker has won this prize alongside Demis Hassabis and John Jumper for their work in protein structure prediction. Both of them were working in a different, the AlphaFold project, but all three of them together. And also, if you look at the papers that have been written, one of the downsides to the Nobel Prize is that only 3 laureates can be named for any category, maximum. Nobel Peace Prize, Nobel Prize for Literature, all those, it's usually a single individual. Science usually happens by collaboration and cooperation. And so in a nod to that, they have over the years given up to 3 people part of the award. And I do think that this acknowledgement of protein design, whether it is the computational aspect, the prediction, or any other aspect, I feel as though I really feel as though it is becoming irrelevant. What do we want to celebrate? Do we want to celebrate these 3 people? Are we celebrating the science? What are we celebrating? And if we are celebrating the science, then we need to find ways to celebrate the entire team of people, interdisciplinary and spread across the world, who worked together to allow many of these discoveries to really take hold. And the teams that are working now are so much bigger than they ever used to be. So big science. It has its, I don't know. What do you all out there think? Do you prefer the lone wolf science iconography, or do you prefer team player where we can all work together? I mean, I kind of feel like team player is going to bring us a better future, but but I
Yeah,
get the
I
lone
like it.
wolfing.
I like it. I
You like
like it.
the
But I like
science
the
rock
team.
star?
But I also want to point out, like, just to be clear, like, what they have done is revolutionized protein design.
Mhm.
But what is designing a better protein used for? All of medicine.
Yeah, exactly. Not just that.
All of the medicine
Yes. Yes. I
are
mean,
protein.
vaccines,
All
so
of
many
your
treatments. Yeah.
industrial biotech
Mhm.
is proteins. All of
Chemistry.
biotechnology
Yeah.
is proteins, right? Like uh, all of discovery of health issues comes down to proteins. Like this at this point, it's all this is the thing that does everything. So yeah, they revolutionized the way that a huge swath of science works.
They
That's awesome.
absolutely.
So yeah,
Yeah.
and of course, there's more contributors than this, but this is a nod from a prize committee to the field. When one person gets it, it's usually meaning that field is being recognized,
But
and
it
they
also,
picked one
but
individual
being,
to ambassador it.
yeah, I mean, you need the ambassadors of the field, but at the same time, the individuals who are receiving these awards are the heads of labs. So we're also acknowledging the individuals who are good at getting funding, who are good at publishing, who are good at, getting their work. I mean, obviously their work is good, or it would not be getting to where it is.
Yeah, which explains why my papers that I have published without
Oh,
funding,
look at that.
and I did completely on my own, and were poorly received, did not receive any recognition.
Mhm. Aaron Anathema is saying perhaps there should be a TWIS award for super smart thinking teams. Maybe we'll have an award next week for some super smart thinking teams, and we'll give them a big thumbs up.
Or at least
Yeah.
we should get more guests now that we have seen the effect of the twist bump.
Oh yeah, absolutely. I
Towards
mean, David
Nobel.
Baker with the TWIS. We interviewed him, so of course, I mean, that led to a Nobel Prize, obviously.
Usually they get the Nobel Prize before trying to climb the mountain and become guests on Twist, but
Yeah.
it can go the other way as well.
Yeah. So let's see. So Demis Hassabis and John Jumper have been involved in AlphaFold 2, and they've predicted the structure of almost all 200 million proteins. It's been used. They made it open source. They hadn't at first, but it's open source. It's been used by more than 2 million people from over 190 countries. And it's helping researchers understand antibiotic resistance, create images of enzymes that can decompose plastics. I mean, among so many other examples. And David Baker in 2003 used 20 different amino acid sequences, the building blocks of life, to design a completely new protein. And they keep doing it over and over and over again.
So I really like where we're at, because so far, our first two Nobel Prize winners created the technology used by the next set. So it is a collaboration. Like all of these are now connected.
It's like
so far.
without machine learning, the second group, the chemists, which is really interesting because if you were to go in order of like how different sciences underlie the other sciences, physics is like more, there's math, physics, and then physics underlies chemistry and because of how everything interacts. And then chemistry underlies biology. So we're going to move on to physiology and medicine now for this last award. Who has won this year? More guys. Okay, Nobel Prize in Physiology and Medicine for this year. I mean, honestly, this field is amazing. It is making tremendous strides, and it has become a huge influencer of healthcare. and potential therapies over the last 20, 30 years. I feel like this is one of the big ones. This is Victor Ambros and Gary Ruvkin for the discovery of microRNA and its role in post-transcriptional gene regulation. MicroRNA is incredibly important for the little tiny messages that occur within the cell. They're not messenger RNA necessarily. They can be. It's not transfer RNA. This is little tiny segments of RNA. They're not genes. They're little bits. They're little bits of stuff that's been copied from DNA, and they regulate genes. So within the nucleus, they don't really usually go out of the nucleus to do other things, but within the nucleus, they spin around and they're like, whoop, I'm going to grab onto this thing. La, la, la. Now do more of that. Do less of that. Ah. So these microRNA are essential for the regulation that is involved in how proteins are expressed. So how blue your eyes are, right? How brown your hair is, or how much melanin is expressed. Those are simple examples, but maybe it's stuff like how well your immune system works because of maybe you have too much interleukin production. And if that's happening, then maybe your microRNA are doing too much upregulation. And how can we use microRNA to regulate essential processes within our bodies for health? How can we use microRNA for, working with plant pathogens? How can we use this? What can we find out about microRNA? It's like the magic inside. It's the special sauce from what I can see.
So I always kind of think of it like the person who's only letting so many people into the club or the theater or the restaurant at a time. The one who's maybe the person who seats you at your table at a restaurant. So he said, point out, proteins are involved in all of medicine. All right, they're doing all of the work for the body. This is like the thing that life is based on. Really, is the proteins. But this is the management structure.
It is. It's like the conductor, right? It's like, well, that's the music on the page, but give me a little bit more bassoons. Give me a little bit less drums. Like, come on, bring it in. It's microRNA is like a little. It's really.
I like that. I like the studio engineer.
Yeah, it's adding the nuance. Sure. The flavor.
This is what's controlling what levels of proteins get expressed and how they operate when they get there. So, and post-transcriptional things too. So it's like the body can pump out anything, but these microRNA are going to determine whether or not they get used.
Yep,
it's very fascinating because this is not a long understood mechanism.
No. And this was discovered in really wasn't discovered that long ago. And it was Victor Ambros and Gary Ruvkin in the late 80s as postdoctoral fellows in Robert Horvitz's lab, who got a Nobel Prize. So basically, if you want a Nobel Prize, go to the lab of somebody who got a Nobel Prize. Might help. But they studied C. elegans and looked at how the gene transcription and translation processes worked in these strains of little worms, looked at how different cell types, what developmental pathways they took and what kind of signals they were getting, that is a new word, getting. And they discovered that there was this gene this LIN4 gene that appeared to be a negative regulator of the LIN4 gene. So how does that happen? What was going on there? So they looked at the mutants. They collaborated. Collaboration, hey science. And they looked at the regulation of this LIN, or actually, no, I'm sorry, it wasn't LIN4 and then also LIN14. And they had, with LIN4, suggestions that it was really short RNA that was causing this issue. And they're like, wait, what? And so then they're like, ooh, but I'm working on Lin-14 now. And so let's look at Lin-14. And oh, yes, why, indeed, it is a short little segment of RNA that is blocking things for Lin-14 as well. Oh my gosh. MicroRNA was discovered 1993. 2 articles in the journal Cell.
But even then, like, this is not an easy. Even today, I mean, it's getting much easier, but it not an easy thing to isolate,
And we're
to identify.
yeah,
and like the sequencing technologies are only in the I say only like recent history. Like I want to say like 8 years. Maybe 10 years that we've really even been able to
I think
figure out what volumes are actually in play to just like this is like checking DNA is one thing. grabbing and sequencing what mRNA are in a sample much more difficult.
Yep. This has been a lot more difficult, but we've now discovered that there are these little tiny microRNAs that are doing all sorts of stuff. And then there are other little tiny microRNAs that are outside of the nucleus doing other stuff. And there's little tiny bits of RNA are controlling our lives, everybody. Little tiny, little genetic dust. It controls.
There was a story
like
last year or the year before where the virulence of COVID was connected to mRNA because
mRNA is messenger RNA. MicroRNA is different.
Yeah. Okay. Well, it might have been microRNA because now I'm confused by this. But it was having the regulatory effect. So it was either preventing. It was if you were healthy and not having extreme COVID type reactions. It was because the RNA were preventing too many killer T cells from being expressed or being allowed into the air. There was protecting tissues by saying, okay, enough killer T cells, the rest of you need to go. But if you didn't have the production of these RNA, there was nothing to keep the killer T cells from coming in great numbers and attacking the body's own tissues. So,
how else are they used?
Really crucial stuff to understand.
Yeah, super crucial. I'm trying to speak tonight on a podcast.
Doing good.
Great. You want to tell me why I'm less pessimistic than a bee?
Well, you might not be.
I got the buzz.
I don't know.
I got the science buzz going on.
This is from Newcastle University. They say stressed bees make pessimistic choices and may experience emotion-like states.
I like the caveat. It's emotion-like states.
Well, it's hard. It's hard to determine emotion in a bee. We can anthropomorphize, but we don't really know how a bee feels. However, we can tell they get stressed, and we can see that they become pessimistic when stressed. The scientists at Newcastle University have found bumblebees have an adverse response after, or have a negative response after an adverse event. So they had trained these bees on different colors, sort of did one of those training models where like, here's the good reward with this color, here's a lesser reward, this color, and the bees learned the difference and visited the more lucrative locations based on color. So we have trained bees now.
Sounds smart.
Once the bees learned this activity, they some of the groups experienced simulated predatory attacks, and another group did not.
What is it? So what is a simulated predatory attack? A research assistant coming in going, attack. Like, or is it, did they have a bug on a stick? What's going on there?
I don't know. I don't know how they simulated
Hilarious. Okay.
the attack. So basically what they found is after this simulated attack, the bees' foraging behavior changed completely. They began going to the less lucrative rewards. And they think it was just they say the best explanation of the behavior is that they expect high rewards to be less likely after the event and exhibit the traits of pessimistic people.
Oh, poor bees. It looks as though their their attack was shaking the bees or the beehive.
Yeah, our research. This is Dr. Olga Prosenko, who's the lead researcher, who was the lead researcher at the Newcastle University, has moved on to a different university. But she says, our research suggests that like other animals, including humans, bees may experience emotion-like states when stressed, as demonstrated by a clear shift towards pessimism. When faced with ambiguity, stressed bees, much like someone seeing the glass half empty, are more likely to expect negative outcomes.
So by using the words optimistic choice and pessimistic choice, the researchers are already, they're going, well, we're not going to say positive or negative. We're going to say optimistic or pessimistic. So there's I'd love to know how they really define the emotion-like status, because really, when you think about it, it could just be an instinctual response that if you have a negative interaction, negative stimulus, even though you found food or wherever you found food, you don't want to go there again because that is negative survival probability, right? It's not something that is beneficial to your survival. So it's not, I'm just going to argue that this is basic neural network computation and not necessarily what we would consider emotion. So, the emotion like nominal, like the terminology, like, I guess it can it can help us get at the basics of emotion and where emotions came from initially, but
yeah. So I would reverse it. I wouldn't concern yourself too much with bees having the emotional state.
Humans.
But it would be more analogous to say that, like, okay, a human who's had a stress or a frightening, or trauma of some sort may not be as reward oriented. That might be more the basis where you can see how human emotions emerge from experience, less so that a bee is gaining an emotional experience from an event.
Yeah, I wouldn't say that the bee is having an emotional experience, but I guess that's the discussion here, right? Is like we're looking at bees.
It also
what does it mean? What does it mean, Justin?
It could also just be that, like, they forgot all of their training.
I don't think so, because it's not the same. The percentage of just forgetting would have been a much higher. And so you wouldn't just say it's some, the percentage is just kind of less, it changes the calculation.
Okay. So then wait, maybe I'm catching up to what you were saying before. So then they had a strategy that they had figured out through learning the game. But because they encountered a predatory event, they are thinking maybe that wasn't the best strategy because I have encountered a predatory event and I hadn't before I learned the game. Yeah,
kind of a game theory, kind of, estimation, probability based factorization. But I guess what we're getting at here is that there is a probability calculation based on previous negative experiences that changes future behaviors because of past. And I think that this is like basic neural network kind of stuff if you're dealing with survival statistics. Like, you know, if you're an organism trying to survive, you're not going to go toward the thing that wanted to kill you. If you do, you die more likely. Yeah, I don't know, but there's so many interesting things with bees because of all the work on what has been called playfulness, which I think is just exploration. Yeah, I don't know. What do we do with this information? Don't stress bees.
No, but if you stress out your workers, they'll be less likely to expect rewards.
Exactly. Okay,
So,
stress out your
yeah.
workers. They'll be more compliant.
Better workers.
Yeah, better workers.
Yeah.
Great.
Sounds great.
Yeah. They'll be the same as glass half empty. Fantastic.
Oh my goodness.
I have really cool story out of the late Cretaceous. Researchers looking at the Hell Creek Formation, Dakotas, Montana, Wyoming, Sioux, the T-Rex, one of the largest complete T-Rex specimens ever found, was discovered in the Hell Creek Formation. Lots of Triceratops, T-Rex, big, people love these dinosaurs. This is where they were found. We've also found some birds. And now a researcher at the Field Museum at the University of Chicago has presented a study based on 3 toe bones, describing 2 new species that are extinct of predatory birds. I think this is so exciting. It's fascinating to me. All right, so 68 million years ago, big bada boom, a lot of dinosaurs and things died. A lot of these predatory birds did as well. They were all in the ground together. And so there's been a bunch of specimens and bones put in boxes in museums and ignored, just collected. And then this researcher, Alex Clark, he even says himself, he's like, yeah, you know that these fossils, when I first saw them, they were toe bones. They weren't that dazzling. They were a foot bone that the toes attached to called the tarsometatarsus. No skulls, no claws, nothing else. These bones were the size of an adult thumb. Like your thumb, my thumb. I mean, our thumbs are probably different sizes, but these bones were about that big. So thumb size. It's fairly a decent size, right? Okay. These are big bones for a bird. And then Alex started looking at them and he went, wait a minute. There's a bump on this bone, a tubercle that's in a place that it is different from most other birds. The only other birds that this tubercle's location is similar to is hawks and owls, birds that typically catch and carry their prey. So this study is an in-depth analysis of the incredible morphology of these little toe bones. Really not much at all, just not many bones at all. These pictures that we have are from Alex Clark, and they describe new species, Avisaurus darwinii and Magnosavis ecalacaensis. And the reason that these new species are described and being considered new species is because Alex is an ecologist as well. And so he was able to think about what forces would be necessary, what birds of modern day these bones were similar to, and imagine the lifestyles of the birds, the sizes of the birds, and how these birds actually would have existed. So they were big birds that carried prey just like raptors today do. So this is a really exciting new discovery. Hopefully they actually do find more bones there than just these toe bones, these tarsometatarsi, and are able to add to our understanding of this type of bird, this family of birds called the enantiornithines. Basically, these old birds that have died out, but were a group, the Avosorids, that predated a bunch of modern birds. So this is a step in understanding how birds evolved. And did these birds look like modern day magpies, but big? Did they look like old dinosaurs? All we know is that they caught their prey from above and carried them away. Because that's what their bones were structured to do, which is super cool.
Yeah. It just dawns on me, if it has a toe bone the size of a thumb, that is a massive taloned.
It would be huge,
bird.
right? And this is like an
But it could be that they just a small bird with really large feet.
Which is less likely. I mean, they went extinct, so you don't really know. But
Yeah.
it's more likely that they were a very large bird, which is kind of exciting. What did they look like? I want to know more about what they looked like. All we know now is that they had very strong feet.
Well, do we have any like ancient bird fossils that are just missing their feet?
Not like this. No,
Like-
no, we don't. Yeah.
I don't know. Sometimes this happens though. We find part of one thing here and a part of another thing over there, and then
Put them together.
eventually figure out, oh, hey, they're connected. They were once part of the same
Hip
creature.
bone's connected to the leg bone, for sure.
All of the bones are not technically.
Yeah, it's kind of fun. More diversity. Long ago, that died. Thanks, Bones.
Mhm.
You want to tell me about diversity? What do you mean, plunging?
Yeah.
We're plunging biodiversity right now?
So the biodiversity of the planet has been declining in recent years, largely due to human activities of global warming, deforestation, urbanization, pest elimination. All the things that we do as humans have an effect of reducing biodiversity around the planet. The biodiversity within humans, our microbiome, has also been declining as domestication, monoculture, agriculture, and the processification of our food supply has increased. Our microbiomes have become less and less diverse. But there are some places on the planet where biodiversity seems to be thriving. One of them is your toothbrush, your bathroom in general.
I knew it.
In a new Northwestern University-led study, microbiologists found that shower heads and toothbrushes are teeming with an extremely diverse collection of viruses, most of which we have never seen before. And while I'm using the ominous voice, the good news is these viruses, they don't care about people. They're there for all the bacteria that we leave behind. That's what they're going after. The microorganisms collected in the study are bacteriophagy, or phage, a type of virus that infects and replicates inside of bacteria. The researchers know very little about them.
We've got lots of bacteria on the toothbrushes as well. But this is the exciting
So we're
thing.
leaving not a monoculture, but we're not leaving, we're leaving probably everywhere, like all humans are probably leaving pretty similar biosigns behind. The bacteria that is likely to grow on skin is pretty common. the things in our mouth are going to be pretty common. So what we're putting out isn't going to be a huge diversity of bacteria, but there is a large biodiversity of the viruses that go after them.
A huge
So we're helping.
biodiversity. Yeah.
We're helping.
What happened?
So yeah, they think that they say this is a quote from the researchers, basically that the previously unknown viruses that are lurking in our bathrooms could actually become what they say, a treasure trove of materials for exploring applications of using using viruses to attack bacteria. Bacteriophages is another form of an antibiotic, after all. So there is, but they're saying also this doesn't mean you should be doing any extra cleaning or anything, outside of using regular soap and water and rinsing things and washing, normal washing. You don't need
getting
to
a new
do an intense
toothbrush.
Yeah, get a new toothbrush once in a while if it concerns you. Yeah. They wanted after characterizing some of the bacteria, they used DNA sequencing to examine the viruses living in those samples, and they comprised a list of more than 600 different viruses. And actually, no two of the samples that they had taken of bacteria came up with the same diversity of viruses. So we're talking about really a huge diversity of virus types.
And how are they spread between the toothbrush and the shower head? Are they all the same? Is it just because it's on my toothbrush, it's also on my shower? Is that the same?
So I don't think I broke it down like that, but usually in those studies, it's the sink.
Mm-hmm.
The drain around the sink is the worst, along with anything else that you use to touch everything and clean, like sponges or towels and what have you.
This is pretty incredible. What we're seeing though is, like you mentioned, we're using phages for medicine now, right? Where our antibacterials are not working any longer. Suddenly we are able to use phages, and the phages are then able to combat bacteria because that's what they do. Yeah. So applying the phages is actually more efficient sometimes than.
And this is also kind of interesting. One of the things they noticed in this collection of viruses was more, mycobacteriophage?
Yeah, the mixes, yeah.
Than other types. And these infect mycobacteria. Mycobacteria is where you get things like leprosy, tuberculosis, chronic lung infections.
That's cool.
So this is this may be a viral immune system outside of the human body.
Yeah.
That we have attracted by leaving behind toothbrushes. Right?
I don't think that's exactly how I would describe it, but I guess you could say that.
I'm not saying all of the benefits of biology are intentional.
Yeah.
Yeah.
Yeah. By putting toothbrushes in our mouths and making places for bacteria to grow into their colonies. Yes, you're doing that all the time, by the way, everyone.
We give a place for the viruses to replicate. And if those viruses that are replicating are the kind that are going to prevent tuberculosis and leprosy, I'm all for them.
Oh, let's do it. Yes. Yes, yeah. Please. I say yes.
I am done washing my toothbrush. That's it.
No. Your toothbrush is not.
No post-brush rinse for me.
No, your toothbrush is not going
I'm supporting my community.
No. You should get it. No. That's wrong.
We need some sort of a banner that we can put up.
No,
It's
no. No, you're thinking about
like
it
a
in the
little bubble
wrong
that comes up,
way. That's the
like a
wrong
disclaimer that's like,
conclusion.
wrong conclusion
Ah.
from the study.
Hey, all you high school teachers out there, ask your students why Justin's conclusion was wrong. All this and more in our TWIS high school student supplementary materials that we do not make, but we should. Does somebody want to do that? That would be rad. Okay, moving on to this next story. We're going from microbes to comb jellies because comb jellies are awesome. You know comb jellies, right?
I'm just picturing a jellyfish right now. I don't know all the jellyfish
Comb jellies are the really cool ones that
mushed
look like
together.
they've got the bioluminescence all down their little veins. Like they're
Yeah.
incredible looking, and they undulate through the water because of all of their magical little flagella, whatever. They're really cool looking. They're not usually very large, but these comb jellies, they look like space aliens in the ocean.
Yeah. These are the ones that never seem real.
No, Blair loves the comb jellies. I'm sorry to bring.
Like they have LED strips. Like, come on.
They are
That can't
nature's
be-
LED strips. I mean, that is that.
Yeah.
Yes, they have, or they've got their own internal disco constantly.
And they can do some kind
Mm-hmm.
of wild shape shifting as well.
Okay, so you mentioned the shape shifting. Yes, they are a little flexible in their conformations. Researchers were just looking at comb jellies in their waters. The University of Exeter and National Institutes of Natural Sciences in Okazaki, Japan. And the researchers were like, hey, we've got these comb jellies. And they're really awesome. And we're really looking at them. And then they're like, what is that one comb jelly doing? It's not like the other comb jellies. And the one comb jelly looked like it was bigger. Like it looked kind of like it was two comb jellies kind of put together. And so then the researchers were like, I wonder what happens. Do they get injured and then fuse with each other? But that would be funny. Ha ha ha. Long story short, the researchers were able to injure by cutting off about a small percentage of comb jellies that they had and pairing them up in seawater tanks together. And 9 out of 10 times, those jellies that had lobes or partial lobes removed fused with other jellies to create a superorganism. Now, it wasn't just like, oh, let me put my arm around you and help you walk, where one jelly was helping the other jelly. Oh, no. They did experiments to see what exactly was happening. So they looked at muscle activity and the nervous systems, and they discovered that within about an hour, they went from kind of random muscle and nervous system activation on either organism as they started to fuse. After a couple of hours, the syncing of the nervous systems increased and increased and increased. And after 2 hours, 95% of the animal's muscle contractions were completely synchronous.
They really became one
within
thing.
2 hours.
Wild.
And then they were like, oh, let's do some like fluorescent dyed food and see what happens to the food. Okay, the comb jelly, one comb jelly would eat it they had 2 mouths and 2 anuses still. Well, one for each organism. Guess what happened, Justin?
I am afraid of the possibilities.
So I'm trying to open an image and it doesn't want to open it because this is not an open access article, which is unfortunate. So I can't get all the images.
My guess is my worst guess.
Yes, your worst guess. Let's go with your best guess.
Well, it might be my best guess also. Is that at the point where you've identified one thing as a mouth, and you have a digestive system that's connected, you could have it going out 3 different places.
Yeah, so it's not 3 different places. But now, because you have a comb jelly with 2 butts, it can poop out both of them. So the fluorescent material would go in one mouth and then come out the other butt. Their guts fused. Their guts fused.
Yeah.
Yes. So it's like that one South Park episode where there was a monkey with 2 butts and then 3 butts. And they just, South Park, that genetic researcher just kept increasing the number of butts on the animals. But here in this study, there were no researchers involved in creating, I mean, I guess they were actually technically involved in creating these butts, but this is natural. This is just what these comb jellies do. They fuse. And the reason that the researchers think this happened is that they don't have what is called allorecognition. Allorecognition is the sense of self. So it doesn't matter what part is whose, as long as the parts are all whole. And so an injury is going to lead to these organisms coming together as in healing, because that merging is beneficial to the whole, as opposed to the lack for the parts. And whereas other organisms would not be able to fuse in this way, you and I could not fuse. We would one of us or both of us would have to take immune system repressive drugs, right? We'd have to be on a chemical of cocktails to allow ourselves to be connected to each other. But these comb jellies they're like, hey, friend, you help me, I help you. Now we have the same gut and 2 butts. Dun, dun, dun. What's fascinating, though, is they don't just fuse, but they actually do fuse in a way that brings their nervous systems together to coordinate activity and also physiological functions. So the gut, the mouth, and the anuses are not necessarily involved in the signals that make the organism go get food or not get food or do whatever it does. The gut, however, is a massive part where the food is going to sit, be digested. That is going to be part of determining the level of an organism's behavior. So in this case, the fusing of the gut and the nervous system means that the organism, not organisms, but the organism can work as a single entity to do whatever it needs to do. Now, the next question is, how successful are these organisms compared to other of their kind, right? If you're a fused cone jelly, is your success better or worse? Is it?
My guess is it's significantly worse. Yeah. Because
I don't know.
And the reason I would immediately jump to that is that if it was more successful, then you'd see more of the fusings. Mm-hmm. You'd see 3. I don't know if there's a limit to this. 4, 5, a super megalithic creature.
And maybe we just haven't seen that because we haven't, comb jellies have not been raised in captivity for an incredibly long time. They're hard to raise in captivity. And I don't know, maybe we haven't been doing it long enough. When you're in captivity, how often do you get injured? I think there are a lot of questions. We haven't seen them in the wild. In the wild, would it just be unlikely for them to all fuse together?
Well, I
Like,
don't.
is it just
My guess
probability?
is it makes them easier to be preyed upon.
Yeah, I would.
I mean, you get a big super
Sea
organism
turtle.
that's
Come eat that.
yeah, and it just makes it like, oh, I see you.
Yeah.
Okay, munch, munch,
Crunch, crunch, munch, munch.
done. It seems like one of those things that because it doesn't have necessarily a defensive system, but is probably existing based on its size and speed, and being bigger isn't always better.
I think that's a really fair point. But it's novel discovery for science, and now it allows us a pathway to look at this evolution of self, non-self recognition, and how that ties into our immune systems, our nervous systems, so many aspects, because cone jellies are related to us. Oh,
I'd love to see
ancient
how many
ancestors.
those they can
Cousins.
get.
I don't know. How many could we put together? It's a comb jelly with 3 butts. No. Okay, back to South Park I go. You're just looking, you're judging me right now. Hey, everybody, this is This Week in Science. Thank you so much for joining us for another episode of This Week in Science. If you want to find out about really cool fusings of science and comb jellies and all sorts of things like this, I hope you come see our episodes every time we live stream them or release them as a podcast or wherever we do. Thank you so much for being here. If you're enjoying the show, please share it with everybody, or actually your best friend. If you haven't shared this show with your best friend, I'm really going to sit down and we need to have a conversation. If you have not shared TWIS with your best friend, why have you not? Come on, let's talk about the science with our friends, okay? We need to expand our friend circle here. Additionally, if you love the show and you want to help support what we do, head over to twist.org and you can click on the Patreon link. The Patreon platform is where people can support us financially with regular donations, $10 and more a month. We'll thank you by name at the end of the show. There are also various levels with different rewards that are sent to you through Patreon. And as well, you can click on the Zazzle link at twist.org. And when you click on the Zazzle link, you get to find out all the cool merchandise that Blair has created throughout the years related to her Animal Corner calendars, really cool stuff. And also just regular TWIS merchandise. So you can shout your love of TWIS to the world. It all goes to support this show, which is not advertiser funded. It's only funded by listeners. It's really a passion project because we love being here with you. We really can't do it without you. So thank you for your support. All right, coming on back. We don't have Blair tonight, so no Animal Corner. No, but we have so much more science to talk about. I have all sorts of stories that I want to bring up. I want Justin to talk about stories to do. Maybe I'll start with Justin and I will go back and forth in this last part of the show. Maybe I'll start with some brain DNA maps. Do you like brain DNA maps, everybody? Why do we need brain DNA maps? What does that even do? Well, researchers at the Salk Institute have just reported their production in Nature, their report of what is the first developmental DNA map for the brain. And in looking at this, it's not just what DNA is active during development, it has to do with DNA modification. So at what point during different stages of development are different genes, different parts of the chromosome modified? How are they silenced? How are they upregulated, downregulated? How is modification a part of how the brain evolves? And once we know this in human brains really well, this map actually is, the implications of it are that the online platform that they have created will, by being available, and it's really not a pretty platform. If I'm going to show you this platform, and you're going to be amazed at all of the amazing visual stuff that you get here. No, it's not beautiful, but what it is a database for the genes, for the modifications, for the different boundaries of domains, chromatin loops, different aspects of how DNA has been modified. And in the summary for this NCBI Geo Expression Omnibus, love omnibus, it's a great word. Says human frontal cortex and hippocampus play key roles in learning and cognition. So this isn't even the entire brain. Representative structures of the 6-layer neocortex and the more ancient allocortex. They investigated epigenomic and 3D chromatin conformational dynamics of the frontal cortex and hippocampus development using more than 53,000 joint single nucleus profiles of chromatin conformation and DNA methylation. We've talked a lot of times about DNA methylation. Those are the addition of methyl groups to the DNA that change the way that the sequences are read so that they get skipped over or opened up and read more likely. Chromatin conformation is more likely going to be the histidine and also these loops that they're talking about. So these profiles are generated for mid-gestational, late gestational, infant, and adult human brains. Now, one of the things related to this is the difficulty of accessing the tissue for this information, because when researchers are trying to create models for the study of the development of the human brain, we're going to put a bunch of cells together. So we talk about, we grew, organoid in a dish. It's almost just like, kind of like, possibly like the development of a regular brain. But is it? How do we know? So now those models that we're looking at when we're putting things in dishes, when we are modifying them as researchers in the lab in dishes, we know whether or not the outcomes of our experiments are quote unquote normal or not. And so this tissue has been taken through donation, which is very hard to come by and incredibly just sensitive and wonderful. And the fact that people have donated gestational tissue and other levels of tissue to allow us this doorway into the sequence of development that takes place in the human brain, so that we have a database now that can be shared like this, where researchers can access it. Right in this database is not pretty, but if you know how to access it, it's beautiful, because it is going to make what you do a lot easier. Kind of cool.
So sort of always drives some of the point that, to me, that Lamarck was right.
Right. We used to debate this all the time, and I was like, no, I don't know what you're talking about. And I'm like, yeah.
But it's both. But it's Lamarck had always thought that, there was a genetic learning of some sort. Like, the reason giraffes have long necks is because throughout their life and through passing on to generations of reaching up for the tall leaves, they grew a longer neck. And it was completely replaced by Darwin's theory of evolution and natural selection, which is the biggest, a big catalyst and driver for evolution.
But it doesn't explain everything, right? That's the
It doesn't explain everything, and it took discovery of DNA, sequencing of genomes, study of those genomes, to get to the point where epigenetics is understood, that actual experiences in a lifetime, the environment affects how genes are expressed. And then that brings us right back to Lamarck as a driver for evolution of that. He obviously wasn't working with the genome, but neither was Darwin. But one of the things that science sometimes does is it finds a new idea and throws out the old one, which is in some ways extremely positive, because this is what differentiates science from a dogma,
Mm-hmm.
right?
Yeah.
That it itself
Yeah.
evolves and changes.
It's not a bad thing for researchers to challenge their own perceptions.
Right. And archaeology is really great at this. I think it's one of the better fields in it, where they have these set ideas of how things came to be and, through the years. And then they find new evidence and go, no, that wasn't right. We have new information now.
Right.
But it's sometimes we run past an idea like Lamarck, who I know is not they don't teach Lamarckian evolution because it's not a thing. It's not well constructed. It's just the concept. But it is, we are seeing that those concepts are part of the actual evolutionary track. And it's, it is epigenetic changes, experiences through an actual lived life do pass on and do influence the evolution of organisms.
Right. The individuals that lead to population level changes that lead to the actual evolution, right? Of the species. So I think what's, really insightful about what you're saying here, though, is, or that we need to think about here, is how those modifications are, when you're looking at brain cells, those are somatic cells, right? They're bodily cells. They're not the cells that are passed on. They're not germline cells. But when an organism starts out with just the complement of chromosomes and those cells start dividing, the chromosomes have already been, there's first silencing. And so one of the two chromosomes in every cell is silenced. And it's random. It's pretty random. But is it random? I don't. There's an interesting imprinting. They call it imprinting, and it's an interesting process. It's been studied for a long time. But that's like the first level of epigenetics, and then but that's carried along, and then beyond that, there are already methylations and chromatin loops and like other things that change how chromatin is read when everything starts to get opened up and transcribed and translated. But then, like you're saying, there's external forces that come in, and that can cause more modifications and shifts. And so there's this interplay between what's already there and then the environment that development is taking place in. And so that in itself is just a fascinating, fascinating thing to be able to start to puzzle together.
And those changes, as far as I understand, it was some of the past research that has shown it in humans, at least, so that these changes from the environment, usually the ones that it will affect, happen pre-puberty.
Mm-hmm. There's a lot, yeah.
So that's sort of an interesting time frame for affecting things that can then be passed on through or connected to a germline, right? Because these what is the study? I can't remember. I always forget if it's a Swedish study or a Dutch study or whatever with the farms and like if they had a time of famine. This is like back in the good record keeping back in like the 1500s or something. These farming communities were over a few hundred years. The grandparents lived in times of famine when they were prepubescent. Their great grandkids were like more likely to get diabetes or like they had these really strong connections from these experiences that they had sort of already been looking at before epigenetics was understood.
But from this, you're talking about the disease states that occur during development, and that's one of the things that this group is really trying to figure out. So, for example, autism spectrum disorders, right? When does that risk for autism really become something that's present in the wiring? How can we intervene in development to alleviate symptoms of autism? How can we help?
That's in development.
Yeah.
But what I'm talking about is like maybe the mechanism that has created, and this is pure speculation.
I love it. You get into the speculation.
But if you were to say that autism had an epigenetic underpinning, what you would also maybe be having to look at then is, what was the circumstance in the grandparent?
Mhm.
Because this is not necessarily something that's just occurring and then manifesting in the developing individual, but something that they have inherited from a grand or great grandparent.
And it depends on the factor, I'm sure.
Developmental influences, right?
Right.
So,
but it's also, with autism, we know that, okay, there's more likelihood of autism with certain factors in the parents at certain time. We talked about microRNA. There are certain little bits of RNA that get packaged with the sperm and with the egg that determine epigenetic factor. They determine the transcription and translation of these genes into proteins. So there's a bunch of stuff in there that is passed, like you said, grandparent to parent, parent to this F1 offspring generation that we're looking at. And where did it come from? How did it start? But like you mentioned, a lot of the disorders that might affect us in our adulthood are getting their start during these early developmental, prepubescent developmental phases. And so to be able to map out the likelihood of certain changes at certain points in time, know where they come in or don't, can help us figure out how to study them and how to treat them.
And what I'm suggesting is, you also need to do a multi-generational study, because the weird thing about that farm study was that it seemed to always skip a generation. It wasn't, if they were connecting environmental things from the grandparents, prepubescent, to the grandchildren's expression of disease risk. And so this is a good step here, but it's also like, you need to look at, you need to have like 3, maybe 4, could be, but maybe those great grandparents were predisposed to something that their grandparents.
So that's that's like getting at like, okay, this is how it's passed down and what's passed down. But like just being able to look at it within, okay, organismal development. What are the places where we can intervene? Yes. What are the spots? If we know that there is something coming down from the grandparents or, family history, what are the places? If we know there's a genetic predisposition, at what point in time do you step in? Because if you don't step in at the right point in time with the right signals, you could be messing everything up. Or you could be doing like just sending it off on like another weird direction that you don't know where it's going to go. So these are all things that need to be looked at. Yeah. You want to tell me a story? You're like, nah, I'm done. No.
So, yeah, I'm good. No, this is I don't know if you've ever heard of Park Run.
No. I mean, I know people run in parks.
Yeah. So, I have noticed that it does exist here in Denmark, but apparently it started 20 years ago in the UK.
And you knew about it 20 years ago?
No. I've only recently encountered it here in Denmark. Like, why are all these humans like standing around getting ready for this big run? But what it is it's a 5-kilometer run. It's kind of a social event. It's free. There's an organization that puts it on. It's called Park Run, and basically they encourage people to get together and do this big run together.
Okay.
The social thing, kind of the sort of annoying thing. If you've ever worked for a big corporation, they might have made everybody say like, "Hey, let's go out and do a fun run." Or a walk and talk.
And we do it here in the US. Usually it's for like fundraising. The 5Ks very often
fundraising for charity.
Fundraising for charity, sort of or it's like hey, this is a health endeavor. Then the HR department gets behind and says, everybody come on out and run. And each department's going to be a team. And people who have never run before run too far and are hurt.
Oh, no.
Because they're peer pressured into taking part into something that they should not be doing. Anyway.
So this parkrun thing has an organization behind it that takes donations, and then they have all these organizers and volunteers that kind of put on the event. It's in 22 countries currently, or more than 22 countries. In the UK, this is a weekly event. In the UK, there are around 200,000 people who every week, now maybe not the same 200,000 people every week, but they get in about 200,000 people a week participating in these runs. And the park run has been endorsed by the World Health Organization. It's a physical activity initiative because according to the World Health Organization, people who are insufficiently active have a 20 to 30% increased risk of death compared to people who are sufficiently active. So there's lots of diseases that people can get. This apparently prevents communicable diseases even by being, somewhat physically active. And as the world becomes even more sedentary, like we already went from hunter-gatherer to farming, where we became much more sedentary in our lives, and then we got all our technology, so we didn't need to leave the house even to go grocery shopping, and so. It's a concern for the World Health Organization.
Mm-hmm.
Anyway, they studied, this is a perfect thing to study how physical activity, or increasing your physical activity can affect your life satisfaction. So they had 548 newly registered runners that they gave an interview to, pretty simple interview at the beginning and then again at the end. Basically a 1 to 10 scale of satisfied, not satisfied at all, completely satisfied, somewhat satisfied, sort of a 1 to 10 scale. Participant responses were grouped based on their self-reported activity levels, their sort of physical activity before they had signed up for this park run.
All right. Yeah.
And the results indicated significant increase in life satisfaction for the normally least active participants. Their group saw their average scores rise to 7.389, a change of 0.768 points. Which doesn't sound like
a lot, but if it's more than the standard, more than the error, I mean, that's good, right?
That was their increase. When they included all the physical groups, the improvement was only 0.257, but still significant. They think that might be due to the fact that some of the more active groups, which was actually most people who are joining these things are already active, physically active.
That's what I was going to wonder. Like, how much is it actually changing people's behaviors? So you have active people going, I get to go running. Awesome. I want to run it. Yeah, that's great. I was going to run anyway. Why not do it with other people? This is awesome.
And it's exactly what they think is happening. Like, they think the reason that there's less increase in life satisfaction for the more active folks is because the more active folks are just replacing something else that they would have done. Like, they were already going for a run, but now they're going to do it in a social way. So they still saw like a little bit of a bump because maybe instead of going on their run by themselves, they've now joined this sort of group activity. And so that's a little bit more fun.
And that could increase, that is going to increase life satisfaction because you have suddenly more of a social life, right? You're laughing and talking with people or, being competitive and tripping people, but whatever. It's all social.
But there's also, when you're saying, like, when we're looking at, 0.768 increase amongst the least active and 0.257 increase, doesn't sound significant, but here's the comparable reference for the same surveys that they were doing during COVID-19, that when the pandemic was in full lockdown.
Nobody was running for a while.
But the life satisfaction on that scale in the UK had moved down, the average for the population at large had moved down 0.39. So the least active runners, the people who are normally least active who've joined this event and stuck with it for the 6-month period, they saw almost like twice the boost than the pandemic lockdown was a negative, right?
Yeah.
Like, so on that, when you
Because
put
they
it in
lost their
terms
social
of,
circle, they lost the running, they lost they lost a connect, they lost so much.
well, they weren't running during these are the least active people. They probably hadn't been running for a long
Yeah.
time. But what the boost that they got from participating in this was almost twice as good as the
drop.
deficit
Yeah.
that the general population felt during the pandemic. So
Oh, okay. Okay.
really,
I see what you're saying.
it's a pretty striking increase. Also, they tried to monetize it because there's this, what is, when you're doing public policy for...
Isn't that always a psychology thing? It's like, oh, well, let's put this in dollars. Everybody understands dollars.
Well, the reason they do it's pretty straightforward. You're going to spend resources from your country's health department. What is the thing that's going to be effective?
Mm-hmm.
How can we positively spend our dollars to improve health?
Avoid
Does it have a
COVID.
yeah. Yeah, we should have been spending those dollars.
Yeah.
But the number was insanely high. Like amongst the so they had this, it's called the Welby scale, well-being adjusted life years, and it tends to equate 1 point in rise or fall in this life satisfaction to a spending to a value to society of 13,000 pounds. This is a UK study, so everything's in pounds. You have to do your own conversion. They found a benefit to cost ratio for these least active participants was somewhere in the range of 98 to 1, meaning if you spent 98 pounds, if you spent 1 pound of incentive to create this program, it was worth 98 pounds in actual returns on that investment.
That's an incredible ROI. Like when you actually think of ROI, nothing. That kind of ROI is great. Yeah. Nothing does that.
And so they put it in the dollars because they're considering spending dollars on health initiatives. And so that's why it's framed like this. It's not that this money is actually being generated itself.
Yeah.
But averaging it across the 2019 numbers of 400,000 participants in the UK over the year. So it's 200,000 about a week that are showing up to these things, but it's twice that in the number of yearly participants in 2019. They said that'd be like 266 million just for a half year of participation in pounds. So the event itself costs 4.5 million to organize. So it might be, adding a social element. The thing is free to join.
Yeah.
There's all the encouragement, and this is I think really important. If you're not an athlete, going on one of these, and you can walk it, you can run it, you can walk it, you can your own pace. It doesn't really. There's no conditions on it.
There we go. That helps.
If you're not the athletic type and you're thinking, that's for the athletes, you're getting way more out of it.
Yep.
You're getting like 3 times more out of that activity than people who are already.
And so I'm going to add something else to this as well. So if you're not physically active, maybe you don't also have a big social group. You don't get out much, right? So you start doing this. You improve your physical health. You also improve your mental health through, the physical health and also the social interactions. And in that, you're also going to improve your immune system's ability to protect you. You're going to be more resilient to stress. All of these things, when we're talking about the life satisfaction, that's one thing. But additionally, for just public health factors, the money that goes into the country paying for these park runs is a drop in the bucket, because what they're doing is actually improving the health of the population and the resilience against future disease. So say there is something, bad flu, COVID, whatever, COVID-like disease, the population who are healthier are going to be more protected. It's already there.
Yeah, and that's exactly the reason for putting those well-being, life satisfaction things in dollar amounts. It's for a government that spends money on its people's health.
Let's do it.
In the United States,
I don't want to talk about it.
okay.
I don't want to talk about that. But you can tell me about really far off stuff, though. You want to tell me about big far off galaxy? That's something.
The researchers have discovered the most distant faraway galaxy that is Milky Way-like. So there's a lot of really far away galaxies. This one, dubbed Rebels 25, is a disk galaxy.
Who named it that?
Seems just as orderly as present-day galaxies. Seems like the kind of galaxy that could be hanging out, in the cluster of galaxies where the Milky Way is. We've had all this time to perfect galaxy being. We're nice and smooth and disc-shaped. Only this one, based on the redshift at the 7.31, is what would that make it? 12.5 billion? More than 12.5 billion years old.
That's old.
It shouldn't be there.
Wait, what?
It shouldn't exist. The just galaxy formation under standard model, the lambda CDM. This galaxy formation is supposed to take, galaxies are supposed to not really even be there for the first billion years. These types of galaxies needed many billions more to get nice and smooth and have everything look nice and disc-shaped. It's too advanced. It's like finding an iPhone in an archaeological site. It's not supposed to be there. So, messy galaxies. That's what we expect. Early primitive, primordial galaxies. Yeah, that's fine. You can have those way back. But one that is ordered as the Milky Way, rotating disk with tidy structures, spiral arms, billions of years this is supposed to take under our current understanding.
So how?
What
do they think? How? Why? What? Is it actually a mirror image? Are we having ourself reflected back at us?
Well, it doesn't look exactly like the Milky Way. Unfortunately,
all right,
we've looked in this mirror and found the other side of the mirror actually seems more developed. Yeah. The data is hinting at more developed features similar to those of the Milky Way.
At that early time point in the galaxy.
Central elongated bar, the spiral arms, they need more observations. But it's like this doesn't look anything like what we would expect. So I'm going to raise this possibility.
I like possibilities. All right.
Because there's also some JWIS has been finding stuff. I think it's a JWIS discovery, at least the Jades galaxies. There's a couple of them now. The different Jades that's in a galaxy cluster that is a couple 100 million years after the Big Bang. The one we're talking about now would be 700 million years after the Big Bang. There's also these Jades cluster galaxies that they're finding 200 million years after the Big Bang is supposed to happen, and there's giant galaxy clusters with galaxies that don't seem to should not exist according to the current things, which means we need to change our current understandings. It's all of the evidence of these early galaxies, and now early galaxies with greater, much finer degrees of galactic evolution is indicating a couple of things. One, our dating system for cosmic objects using redshift needs help. It's not as accurate.
Might need some help. Yeah.
The problem is, if that's not accurate, then we don't know how old any of the things we've looked at are, or even how to compare them, because that's primarily what we're using to age.
But that's what the interesting, like when people, we've talked about it before, and it keeps popping up, and I wonder about the idea that the universe is not homogeneous, and so there are potentially spaces where is the speed of light faster or slower? Are there spaces where the warping of space-time is different in a way that we can't detect usually? There are questions about certain aspects of how we measure the universe and everything in it, and we take our position in what we see, we have to take with a grain of salt, right? We don't know everything that's between us and there. We have guessed, we have made assumptions, we have made inferences, and we're doing the best we can, and our resolution's all right, but we're still missing a lot. And so it makes me wonder if, like, okay, maybe some places that redshift, maybe what we measure as 7 point whatever, it's not the same as some other area of space for whatever reason. And I'm just throwing it out there because it has popped up a few times over the last few decades. And I think that it's not a proven hypothesis by any means, but I think it's just something to consider,
okay.
right?
I'm gonna pause it and say, actually, you're right. It is two things. One, it looks like the universe is very homogeneous in terms of how physics and redshift work, and partly that's because as we go looking into what was the DESI project that grabbed light from like a million galaxies. They've got this really beautiful site you can kind of fly through artist renditions of their galaxies. But when you go back and you look at regions of space, it's very consistent, like large. But we're also, whenever we're talking about large regions of space with millions of galaxies, we're talking about taking a coin and holding it up to the stars and saying we've looked at less than this, right? But one of the things it did was it found there's this thing called the Hubble tension, right? This is the accelerating, expanding universe. Things way in the distant past seem to be accelerating at a slower rate than things that are nearby that we've looked at in terms of how much their redshift is changing with time. That's the idea that not only is the universe expanding, but that rate of it is speeding up. And it's called the Hubble tension. It's part of why we see a very big difference in distant galaxies' rate of expansion than closer galaxies. The DESI project kind of came up with more data because that data is a lot of it is from the 90s, I think, from some of the earlier ways of looking at redshift and calculating rates of expansion. DESI kind of put a number that's closer. So now it more looks like the universe is expanding, but maybe not so much accelerating like we were thinking. Like maybe that rate isn't getting quicker. There isn't a big difference in it looks more homogeneous anyway than it was before. However, even with that, we're having to create dark energy, dark matter, and a whole bunch of things to explain rotation of galaxies far away, to explain, rotations of spiral arm galaxies that should be flying apart because of what a standard model has suggested must be in place. We're seeing what looks like less of a dark energy signal in that DESI model than we would expect, meaning we would actually be maybe, according to that, maybe even heading to a contracting universe from a period where they're looking back, billions and billions of years. There is something wrong, certainly, with our understanding of the age of the universe that Jay Wist is pointing out. There's obviously something problematic with discrepancies in redshift. Overall, it's highly uniform, but there's weird things that keep showing up. Like there's a tendency for galaxies that have rotations, I think, that are the same as ours.
There's a lot.
To have different redshifting than ones who are rotating opposite us. And those local co-moving redshifts of things moving around should almost be invisible compared to the amount of redshifting that's been attributed to the expansion of space.
Right.
So if there's things that are and have different rotational angles, and that is being amplified through redshift, it means this whole expansion cannot then explain why that's there, because you shouldn't see any signal. And the fact that you are seeing signals from some of these also says maybe our expansion based on redshift alone is not completely accurate. So
there's a lot.
There's a whole lot of questions.
There's a whole lot of questions. But
we need to get somebody from that DESI project, or maybe somebody who's analyzed it.
Well, that would be a great thing to do. Maybe we will. But next week we're going to have Ethan Siegel, who's he's a great physicist. Starts with the bang.
The karate guy?
No. We've had him on the show before. Maybe it was when you weren't on. But
we had Steven Seagal on the show before. I don't think I must have been off that week.
Siegel, and he starts with the Bang podcast and blog and other things. And he has a new book about the James Webb Space Telescope out with National Geographic. And so we'll be talking with him next week about James Webb and looking at the universe and physics and lots of cool stuff. And so maybe you can talk with him about redshift. We could do that. But he's not part of that whole DESI cabal. All right. My last two stories tonight. First, let's talk about astrocytic autophagy. Dun, 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 dun. Woo-hoo. Let's dance in the minefield of the brain. That's right. Researchers just publishing in the open access journal Molecular Neurodegeneration. They have published their study looking at how astrocytes deal with amyloid beta proteins in the brain. This is a group out of the Korea Institute of Science and Technology Brain Disease Research Group, also in collaboration with researchers at the Institute for Basic Science and also Boston University Chobanian and Avedisian School of Medicine. Lots of international collaboration going on here. But the super cool aspects of this study, the things that I think are just the coolest, have to do with what we already kind of knew about astrocytes. So astrocytes are like the garbage trucks of the brain. They're support structures, but they're also involved in, things are getting messed up. We're part of keeping everything cleaned up, happy, and doing all the work to clean up the dirty muck. And so when they're working normally, as they do usually when you're young, everything gets cleaned up. Your brain is free from debris and everything works properly. Dun, dun, dun. But sometimes you have...
Wait, and how do I get there?
Right. Once upon a time with not much inflammation and not genetic disorders or infections that spur on disorders that lead to stuff like this. So anyway, the amyloid beta plaques and proteins are known to be involved in Alzheimer's disease. And yes, there are questions about how the amyloid beta plaques are actually involved and which ones are. And there are a bunch of questions about research that has gone on for many years as a result of some retractions over the last few years. But there's still a lot of research that is like, okay, amyloid beta is these plaques. When you have them, you're more likely to have dementia, Alzheimer's disease, et cetera. What's going on? Well, this study, they pretty much they set up astrocytes and did genetic signaling, knock in and knock out. So adding genes in, taking genes out, turning genes on, turning genes off. They did this knock in, knock out to turn on astrocytes to rev them up so they worked really well, or to turn them off and to see what happened. And what they determined is that when you turn down certain genes within the astrocytes, you have a lot more of the kinds of proteins and other things that start to accumulate when you start breaking down. There's a lot more garbage. There's a lot of the metabolic detritus around. And among them, the amyloid beta plaques. And so the researchers were like, okay, what happens when we fix that? What do we do? And how are the amyloid beta plaques involved in this? And in the end, what they were able to put together is that this system is very, very connected, and that the astrocytes are responsible for degrading these amyloid beta proteins, the oligomers. And if they don't, you have accumulation of the amyloid beta plaques. The amyloid beta plaques lead to neuronal damage and decreased memory function. You don't want that to happen. And this is really one of the first studies. People have been studying this for a while, but this is one of the first studies that has really done this work in live animals, been able to actually show the accumulation of the plaques and the damage within the hippocampus of the brain and other areas of the brain, so that there is a link between actual changes in the astrocytes, actual physical accumulation of amyloid beta, or cleaning it up, and then the resulting behavioral changes. And not only that, but the genetic targets that were involved. So, maybe, and we've talked a while about how more metabolic aspects of the brain might be the things that are involved in Alzheimer's disease and some other dementia-related diseases of neurodegeneration and aging of the brain. There's normal aging of the brain, which, as we know, you have slowing down of mitochondria, you have slowing down of brain, different things. When the garbage stops getting taken out, things don't work anymore. This is just more proof of that. You got to take out the trash, everybody. The brain too.
Oh, that reminds me. Thank you.
I hope you make your trash day, everyone. That was your reminder. Last story for the night. I just want to put a final chapter on this evening. Researchers have long talked about how we pay attention to the world, that maybe we go through our day kind of looking at the world like a book, and like our brain taking the pages of the book splits it into chapters. But is it that our brain reacts to the information or the changes that we go through in environments and emotions and other things during the day? Is our brain reacting, or is it top-down controlling the creation of those book chapters? What do you think, Justin?
I don't know.
I don't know. Yeah, well, I do. Well, no, I don't know for sure. But these researchers, they took a bunch of people and they gave them basically story situations. They gave them scripts, a narrative, and they had some people pay attention to certain aspects of the narrative, and then other people pay attention to different aspects of the narrative, and then looked at the parts of the brain that were active during these periods of time, how the brain was timing and signaling and moving things around and creating, basically chunking the story. So this isn't exactly the same as maybe the quote unquote chapter creation for different aspects of you as a person going through your day. But by telling the participants what to pay attention to, so your situation, a narrative, a script related to a restaurant scene. The restaurant scene involves you going to the restaurant with somebody. You're going to the restaurant and, oh, you have to order the food. And then there's the eating of the food. But there are other things to pay attention to. Are you paying attention to just the ordering of the food? Are you paying attention to the interactions with other people? Are you paying what parts of the script have the researchers told you to pay attention to? And what they discovered is that when they told people to pay attention to, say, ordering the food versus eating the food, which do happen at different times of the script, they did see that the ordering of the food caused a neural event boundary. And that's what they're calling it, a neural event boundary between ordering the food and then the chapter of eating the food. And when they told people to pay attention to the eating of the food, that was where the chapter began. That neural event boundary was created based on top-down control and not just the information that was coming in through that script or the narrative that they were listening to. And then to really test it, they were looking at, a question of ordering, going to the grocery store and going and getting your groceries at the same time that you are also finishing and putting together, finalizing a business deal. And so depending on what aspect of the story, of the script, people were told to pay attention to, even though in the script these things were happening kind of simultaneously, it did create separate neural event boundaries based on what the important information was that was being paid attention to. So the idea is that perhaps we have this top-down control, and depending on what we decide to pay attention to, what we are focused on, and really, want to put our attention to, that maybe that will determine how our memories are created and how we remember things. So if you want to remember something in a particular way, if you want to remember particular details, that focusing on those things is going to make a difference. We are not just subject to our environment. It's not just because you put a menu down that the book is closed. Yeah. So with that,
I'm trying to like picture how I would my mental state ever would fit into this. I don't see it.
Well, it's, how, when you're paying attention, when you're multitasking, what are you really paying attention to, right? And so a big question here is, additionally, how we construct our understanding of our lives and reality if we are overstimulating ourselves, if we're trying to pay attention to too many details at once. I mean, some people are really, we can pay attention to a lot of details. That's possible. But our memories and the actual ability to recall, to remember, to learn, that's going to be impacted by what we're paying attention to and how we're paying attention to it as we go through our lives. Yeah. So anyway, on that, I think we're going to call that the final chapter of the show.
Have we done it?
I think we've done it. Have you done it?
Yeah.
Yeah.
I haven't done it.
Yeah. So you can make your memories. Where are you listening to TWIS? Is this the first chapter of your day? Is this the last chapter of your day? Are you going to say, I remember TWIS because I'm by the library? Or are you going to remember TWIS because I just had a conversation with somebody? Or because Justin just talked about something very exciting, a mirror universe? No, not a galaxy far, far away. Red shifting and all sorts of things. We're done here. Are you done?
I'm good.
Okay. I want to say thank you to all of you for joining us for another episode of This Week in Science. Thank you so much for all of your attention and for being here for another couple of hours, and definitely not a tight 90 yet again. Oh, Kyle Pettit is going to bed literally the second that I hit the end button. So I'm going to draw this out. No, I'm kidding. We'll get through this right now. Hey, everyone. I have to give my shout outs, not just to everybody in the chat rooms who are listening live, watching live right now at this very moment, but to a few special people. Vlada, thank you for being patient with me through all of my difficulties. I'm really trying. I really am. And thank you for helping with social media and show notes for the show every single week. You really do an amazing job for the show. Gord, thank you. Arnlor, thank you for being there in the chat rooms and helping to make sure that things are civil and nice, compassionate. Make it nice places to be, actually, right? Identity 4, thank you for recording the show. Rachel, thank you for editing the show. And would we be remiss if I did not thank our Patreon sponsors? Thank you to Eden Mundell, Alan Viola, Aaron Anathema, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, George Corris, Pierre Velazarb, John Ratnaswamy, Chris Wozniak, Bigard Chef's Dad, Donathan Styles, aka Don Style, Ali Coffin, Reagan Shubrew, Sarah Forfar, Don Mundes, PIG, Stephen Alberon, Daryl Myshak, Andrew Swanson, Fredis 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hassenblow, Steve Leisman, aka Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Remy Day, G. Borden, Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Phillips, Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neber, Eric Knapp, Lon Makes, EO, Adam Mishkon, Kevin Parachan, Aaron Luthen, Bob Calder, Marjorie, Paul D. Disney, David Zimmerly, Patrick Pecoraro, and Tony Steele. Thank you for all of your support on Patreon, and I will get the podcast feeds fixed for Patreon ASAP. That means this week. Okay. I'm sorry, they are broken, and I have not fixed them yet. And beyond that, oh boy, we really do appreciate all of you supporting us on Patreon. And if any of you out there would like to join us on Patreon and you have not yet, head to twist.org and click on that Patreon button. On next week's show, we're going to be speaking with Ethan Siegel of It Starts With a Bang fame about his new book with National Geographic about the James Webb Space Telescope. So don't miss it.
Yeah. Hey, when is that? What time is that? Is that going to be the same time as this time?
We'll
or is it a
be
different time?
back.
8 p.m. Pacific time, broadcasting live from our Twitch and our YouTube and our Facebook and our any other channels we might be on.
That's right. But if you want to listen to us as a podcast, be sure to look for This Week in Science wherever you find your podcasts. And if you enjoyed the show, please get your friends to subscribe as well.
For more information on the stories that we talked about, we have links at a website, www.twist.org, so you can dive in further, read the research papers behind the stories, see if you agree with the methods, the methodologies, the results, the interpretations, the analysis. You can see it all. It's all there.
Yeah. And if you really, 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 really love that and you want to talk to us about it, well, you can send us your feedback. And if there's a topic you want us to cover or address, a suggestion for an interview, please let us know on one of our social media accounts. We are out there usually on our personals or under @TwistScience, wherever it is. And you can send us an email as well. I think that our emails are out there somewhere in the ether. Just put twists in the subject line when you reach out so that your email doesn't get spam filtered into an astrocyte who has been super powered by genetic knock-ins to eat all of the detritus.
Cannot wait to discuss science with you again next week. If you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. Science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.